హాయ్ అండి. ముందుగా అందరికి వెల్కమ్ టు మై ఛానల్. ఈరోజు మనం 9th క్లాస్ టెక్స్ట్ బుక్లో నుంచి సెల్ స్ట్రక్చర్ అండ్ ఫంక్షన్స్ నుంచి ఎంసీక్యూ తయారు చేసుకున్నామండి. ఇవి సెంట్రల్కి మళ్ళీ టీఎస్ టెట్ అండ్ డిఎస్సికి ఉపయోగపడుతుందని టెక్స్ట్ బుక్కేనండి, మొత్తం మెటీరియల్స్ కాదు. అలాగే ఈ టాపిక్ ఉన్న ప్రతి ఒక్కరికి యూజ్ అవుతుందండి, ఏపీ వాళ్ళకి కూడా. ఫస్ట్, ద సెల్ మెంబ్రేన్ ఈజ్ ద కవరింగ్ ఆఫ్ plant cell only, animal cell only, both plant and animal cells and bacteria cells only. Option B, animal cell. In a plant cell, the layer present over the cell membrane is called cytoplasma, cell wall, nucleus, vacuole. Cell wall. The plants, in a plant cell, the layer present over the cell. The cell wall is a present in the cell wall అనేది animal cell. Option B, plant cell. Option C, all living cells. Option D, only bacteria cells. Plant cells. Which structure is outermost in a plant cell? Cell membrane, nucleus, wall cell, cytoplasma. Cell wall. Which statement is true? Animal cells have a cell wall. Option B, plant cells have only a cell membrane. Option C, plant cell have both cell membrane and cell wall. Option D, cell membrane is absent in animal cells. Plant cell have both cell membrane and cell wall. Cell wall is a living part of the cell, dead part of the cell, protoplasmic structure, fluid structure. Cell wall is a dead part of the cell. The cell membrane mainly functions as food storage. Option B, respiration. Option C, photosynthesis. Option D, covering of the cell. Cell membrane mainly function covering of the cell. When salt solution is added to the peel of Rio leaf, what happens to the cell? Cell walls, cytoplasma shrinks, cell wall breaks. Option D, nucleus enlarges. Cytoplasma shrinks. The shrinking of cytoplasm along with cell membrane is due to entry of water into the cell, loss of water from the cell. Option C, breaking of the cell wall. Option D, increase in salt concentration inside the cell. Loss of water from the cell. Separation of the cell membrane from cell wall is observed because cell wall dissolves. Option B, cytoplasm absorb water. Option C, water moves out of the cell. Option D, protein breakdown. Option C. Cell membrane is mainly made up of carbohydrates and water. Option B, lipids and proteins. Option C, cellulose and starch. Option D, proteins only. Lipids and proteins. In animal cells, the outermost layer is cell wall, cytoplasma, cell membrane, nucleus. Animal cells, the outermost layer is cell membrane. The cell membrane mainly made up of lipids and proteins. Another name for cell membrane: nuclear membrane, cell wall, plasma membrane, tonoplast. Cell membrane, another name, plasma membrane. The structure of the cell membrane can be clearly observed using simple microscope, compound microscope, electron microscope, hand lens. Electron microscope. Cell membrane is mainly made up of lipids and proteins. One major function of the cell membrane is photosynthesis, protein synthesis, maintaining internal balance of the cell, storage food. Maintaining internal balance of cell. The cell membrane allows substances to pass through it freely, not at all selectively, randomly. Selectively. The cell membrane allows only certain substances to pass. It is called impermeable membrane, fully permeable membrane, semi-rigid membrane, selectively permeable membrane. Option D. The selective permeability of the cell membrane helps the cell to break down food. Option B. Control exchange of substances. Option C. Increase cell size. Option D. Produce energy. Option B. Control exchange of substances. What is the main function of the cell wall in plant cells? Shape and protection of the cell. The cell wall helps plant cell to withstand changes surroundings medium because it is completely rigid. Option B. Exert inward wall X pressure. Option C. Produces cytoplasmic pressure. Option D. Is made up of proteins. Exert inward wall pressure. Cell wall is mainly composed of proteins, lipids, cellulose and starch. Cellulose. Which of the following is not a function of the cell? Provide mechanical support. Option B. Protect the cell. Option C. Maintain cell shape. Option D. Control all metabolic activities. Option D. మిగతా అన్ని కరెక్ట్, ఇది కాదు. Plant cells can tolerate more osmotic pressure than animal cells because they have large vacuoles, they have flexible cell wall, their cytoplasm is dense, their nucleus is bigger. Flexible cell wall, they have a flexible cell wall. Which layer lies outside the cell membrane in plant cells? Plasma membrane, cytoplasma, cell wall, nuclear membrane. Cell wall. The porous nature of the cell wall helps protein synthesis, exchange of materials, energy production, DNA replication. Option B. Exchange of materials. The nucleus was discovered by Hooke, Robert Brown, Swan, Virchow. Option B. Nucleus was discovered by Robert Hooke. The nucleus is called the control room of the cell because stored food, control all cell activities, produces energy, protect the cell, controls all cell activities. Which of the following cells do not have a nucleus? Sick cells, onion peel cells, mammalian or red blood cells, roots, air cells. Option C. Genetic material of the cell is present mainly in the cytoplasma, cell membrane, nucleus, vacuole. Option C. Nucleus. The membrane that surrounds the nucleus is called plasma membrane, cell wall, nucleus membrane, tonoplast. Option C. Nucleus plays an important role in respiration only, photosynthesis, cell division, digestion. Cell division. On the basis of nucleus, cells are classified into prokaryotic and eukaryotic cells, plant and animal cells. Option C. Living and non-living things. Option D. Unicellular and multicellular cells. Prokaryotic and eukaryotics. Prokaryotic cells are those which have a membrane bound nucleus, do not have membrane bound nucleus, have a many organelles. D. Are found only in plants. Option B. దీనికి న్యూక్లియస్ ఉండదండి, ఇది ఓన్లీ బ్యాక్టీరియాస్ లో ఉంటుంది. Which of the following is a prokaryotic cell? Plant cell, animal cell, bacterium, onion peel cell. Bacterium. Cyanobacteria belong to which category? Eukaryotic cells, plant cells, prokaryotic cells, animal cells. బ్యాక్టీరియా అంటే ప్రోకియారోటిక్స్, దీనికి సెల్స్ న్యూక్లియస్ ఉండదు. సెల్స్ హ్యావింగ్ మెంబ్రెయిన్ బాండ్ న్యూక్లియర్ మెటీరియల్ ఆర్ కాల్డ్ ప్రోకియాటిక్ సెల్స్, బ్యాక్టీరియా సెల్స్, యూకారియోటిక్ సెల్స్, వైరల్ సెల్స్. Option C. Cytoplasma is fluid inside the nucleus, fluid outside the cell membrane. Option C. Solid part of the cell. Option D. Fluid between the plasma membrane and nucleus. ఆప్షన్ డి అండి. సైటోప్లాస్మ్ అనేది న్యూక్లియస్ తప్ప మొత్తం న్యూక్లియస్ చుట్టూ అంతా ఉంటుంది అనమాట. అదొక జెల్. సైటోప్లాస్మా కంటైన్ ఓన్లీ న్యూక్లియస్, ఓన్లీ క్రోమోజోమ్స్, ఓన్లీ సి సెల్ ఆర్గనెల్స్, డి ఓన్లీ సెల్ వాల్. ఆప్షన్ సి సెల్ ఆర్గనెల్స్. Which organelles are absent in prokaryotic cell? Ribosomes, cytoplasma, cell membrane, membrane bond organelles. Membrane bond organelles. Ribosomes in prokaryotic cells are membrane bond absent, present in membranelles. Option D inside nucleus. Option C present and membranelles less. Protoplasma was earlier believed to be cell wall, life fluid, nuclear membrane, vacuole. Option B life fluid. Protoplasma is now divided into cell wall and cell membrane, cytoplasma and nucleoplasma, nucleus and vacuole, plastid and mitochondria. Option B cytoplasma and nucleoplasma. Nucleoplasma is substances outside the nucleus, fluid inside the nucleus, fluid inside vacuole. Option D cytoplasma. Option B fluid inside nucleus. Endoplasmic reticulum was observed using electron microscope. The network of membrane present within the cytoplasm for transport of substances is called Golgi apparatus, endoplasmic reticulum, lysosome, vacuole. Endoplasmic ER. Endoplasmic reticulum mainly helps in photosynthesis, cell division, transport of materials inside the wall, storage of food. Transport of materials inside the cell. Endoplasmic reticulum is made up of membrane bound tubes and tubeless fibers, granules, cell wall materials. Option A membrane bound tubes and tubeless. The membrane of endoplasmic reticulum is similar to cell wall, vacuolar membrane, nuclear membrane, plasma membrane. Endoplasmic reticulum having ribosomes on its surfaces is called smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi ER, nuclei ER. Option B endoplasmic reticulum having ribosomes on its surface ఎలా ఉంటుందంటే rough endoplasmic reticulum. Smooth endoplasmic reticulum is defined by the presence of DNA, presence of ribosomes, absence of ribosomes, presence of chromosomes. Absence of ribosomes. Rough endoplasmic reticulum is the site of protein manufacture, lipid synthesis, detoxification, respiration. A protein manufacture. Smooth endoplasmic reticulum helps in the manufacture of enzymes, starch, proteins, lipids. Proteins and lipids manufactured in ER are destroyed immediately, stored permanently, transport different parts of the cell, released outside the cell. C. Transport different parts of the cell. Proteins and lipids manufactured in ER, endoplasmic reticulum, transport to different parts of the cell. In liver cell, smooth endoplasmic reticulum plays an important role in photosynthesis, protein synthesis, cell division, detoxification of drugs and poisons. Option D. Golgi body was first observed by the Camillo Golgi in the year 1839, 1831, 6065, 1953, 1898, 1898. Golgi apparatus can be clearly observed only under simple compound electron hand lens, electron microscope. Golgi body is mainly composed of several membrane forming sac-like structure. Option B. The sac-like structure of Golgi apparatus are surrounded by chromosomes, vacuoles, fluid-filled vessels, mitochondria. Option C. Fluid-filled vesicles. Proteins produced in ribosomes reach the Golgi body through the nucleus, vacuoles, lysosomes, vesicles. Option D. The main function of Golgi body is to produce energy, pack and modify substances, digest waste, control the cells. Pack and modify substances. From the Golgi apparatus, substances are transported to only nucleus. Option B, cell membrane or lysosomes. Option C, only mitochondria. Option D, only vacuole. Option B, cell membrane or lysosomes. After reaching the cell membrane, Golgi products are mainly destroyed, stored, permanently secreted from the cell, converted into the DNA. Secreted from the cell. Golgi apparatus also helps in the photosynthesis, cell respiration, respire and regeneration of the cell membrane, cell divisions. Repair and regeneration of cell membrane. Golgi bodies are found in large membrane in cells that stored food, do not secrete substance, secrete hormones and enzymes, perform photosynthesis. Option C, hormones and enzymes secrete. Lysosomes were discovered in large vacuoles, tiny particles visible in the cytoplasma, cell wall, structure, nuclear bodies. Lysosomes are discovered tiny particle visible in the cytoplasma. Lysosomes contain genetic material, respiratory enzymes, destruct enzymes, photosynthesis pigments. 67. Respirator enzyme. The enzyme present in lysosomes normally fix freely with the cytoplasma, damage the nucleus, come in contact with all organelles, do not come in contact with the rest of the cell. Do not come in contact with the rest of the cell. ఇక్కడ సేమ్ ఉందండి. ఇక్కడ క్వశ్చన్ ఒకటి రాసుకోండి. మెటీరియల్స్ దట్ నీడ్ టు బి డిస్ట్రాయిడ్ ఇన్ ఏ సెల్స్ ఆర్ మెటీరియల్స్ దట్ నీడ్ టు డిస్ట్రాయిడ్ ఇన్ ఏ సెల్స్ ఆర్ ఏంటిదంటే ఆన్సర్ ట్రాన్స్పోర్టెడ్ టు లైజోజోమ్స్. 69 ఏంటిదంటే మెటీరియల్స్ దట్ నీడ్ టు బి డిస్ట్రాయిడ్ ఇన్ ఏ సెల్స్ ఆర్ ఆన్సర్ ట్రాన్స్పోర్టెడ్ టు లైజోజోమ్స్. The main function of lysosomes is protein synthesis, digestion of cellular waste. Option C, energy production. Option D, cell division. Option B, digestion of cell wastage. Sometimes lysosomes burst and repair the cell, stop enzyme action, release enzyme and to digest the cell, produce new cells. Option C, release enzymes to digest the cell. When lysosome digests the entire cell, the process is called photosynthesis, respiration, autolysis, transpiration. Autolysis. Lysosomes are known as suicide bags because they store food, help in secretion, control the nucleus, can destroy the cell itself. Option D. Can destroy the cell itself. Lysosomes help the cell mainly growth, digestion and cleaning, movement and reproduction. Digestion and cleaning. Lysosomes are most abundant in cells that having high metabolic activity. Mitochondria are generally found scattered in the nucleus, cell wall, cytoplasma, vacuole. Cytoplasma. Mitochondria are usually large and flat, small, spherical or cylindrical, square shaped, thread like only, small, spherical or cylindrical. The length of a typical mitochondria is about 20 to 30 microns, 10 to 15 microns, 2 to 8 microns, 0.1 microns. 2 to 8 microns. The width of the mitochondria is approximately 2 microns, 0.5 micron, 5 microns, 1 micron. 0.0 micron. A mitochondria is about how many times smaller than the nucleus? 10 times, 50, 100, 150 times. న్యూక్లియన్ కంటే 150 టైమ్స్ స్మాలర్ గా ఉంటుందంట. On an average, each cell contains about 10 to 20, 50 to 60, 100 to 150, 500 to 600. 100 నుంచి 150. Under a compound microscope, mitochondria appear as a large scale green plates, oval or cylindrical dots, thread like a fiber. Oval or cylindrical dots. The detailed internal structure of mitochondria can be clearly seen under electron microscope. The outer covering of mitochondria is called inner membrane, cell wall, outer membrane, outer membrane. The folds of the inner membrane of mitochondria are known as matrix, ribosomes, vesicles, cristae. Folds 85. Crista. The fluid-filled inner space of mitochondria is called cytoplasma, matrix, vacuum, nucleoplasma. Matrix 86. The mitochondria has how many membrane? 1, 2, 3, 4. 2. Option B. The diagram of mitochondria showing the typical cells is real and exact, imaginary only, hypothetical, incorrect. Option C. Hypothetical. Mitochondria are best described as storage centers, digestive bodies, powerhouses of the cell, control centers. Powerhouses of the cells. Mitochondria are absent in the plant cells, animal cells, prokaryotic cells, eukaryotic cells. Option C. ఎందుకంటే న్యూక్లియస్ ఉండదు కాబట్టి. The scientist who first generated that all plants and animals are made cells are Robert Hooke, Leeuwenhoek, Schleiden and Swan, Virchow and Hooke, Darwin and Lamarck. Options B. Schleiden and Swan. The credit for propounding the cell theory goes to Robert Hooke, Rudolf Virchow, Schleiden and Swan, Louis Pasteur. Option C. Robert Hooke first observed cells in the year 38, 1855, 1665, 1898. 1855. There was a gap of about how many years between the Hooke's observation and formulation of cell theory? 200. Schleiden and Schwann cells theory did not explain structure of cell, function of nucleus, how new cells are formed, types of cells. How new cells are formed. Who explained that new cells arise from pre-existing cells? Robert Schleiden, Schwann, Rudolf Virchow. Option D, Rudolf Virchow. Rudolf Virchow proposed his modification of cell theory 1838, 1855, 1665, 1898. 1858. Virchow modified the hypothesis. Hooke, Leeuwenhoek, Schleiden and Schwann, Darwin. C. Schleiden and Schwann. According to modern cell theory, all living organisms are composed to tissue only, cells and product of cells, organ only, molecules only. Option B, cells and product of cells. The second cardinal principle of cell theory states that cells contain nucleus, cells perform respiration, all cells arise from pre-existing cells, cells have membrane. All cells rise from pre-existing cells. Cells theory is applicable to only plants, only animals, only unicellular organism, all living organism. All living organism. The father of cell biology. Robert Hooke, Albert Clade, Schwann, Virchow. Albert. Anton van Leeuwenhoek lived during. Option C, 1632 1723. Anton van Leeuwenhoek constructed a simple microscope. Leeuwenhoek observed protozoa from blood, rainwater, seawater, pond soil, rainwater. Verticella was observed by Hooke, Schwann, Leeuwenhoek, Brown. Leeuwenhoek. Bacteria were observed by Leeuwenhoek from pond water, soil, his mouth, air. His mouth. Robert Hooke discovered cells in onion peel, cork, leaf, root. Cork. Robert Hooke discovered cells in the year 1665. Robert Hooke used a simple microscope, electron microscope, compound phase contrast microscope. Option C, compound microscope. Robert Hooke used a compound microscope. Hooke observed cells in cork and living plant tissues. Hooke observed cells in cork and living plant tissues. The nucleus was discovered by Hooke, Purkinje, Robert Brown, Virchow. Option C, Robert Hooke. Nucleus was discovered in 1665, 1831, 1839, 1855. 1831. The term protoplasma was coined by Schwann, Schleiden, Purkinje, Virchow. C, Purkinje. Protoplasma term was coined in Option B. 1839. Cell theory was proposed in 1839. Cell theory was proposed by Hooke, Brown, Waston, Crick, Schwann and Clade and Ruska. Option C. Schwann studied plants, fungi, animals, viruses. Fungi. Cell theory states that all living organisms are made up of cells. Who explained cell division? Hooke, Virchow, Brown, Rusk. Cell division, Virchow. Rudolf Karl Virchow explained cell division in 1855. Omnis cellula e cellula was proposed by Virchow. Transmission electron microscope was built by Claude, Rusk, Waston, Margulis. Option B. TEM, transmission electron microscope, was built in 1931. The electron microscope uses light rays, sound waves, electron beams, X-rays. Electron beams. DNA double helix structure was announced in 1931, 1839, 1953, 1974. C. 1953. DNA structure was proposed by Schwann, Sladen, Waston and Crick, Cloud and Rusk, Hooke and Brown. 7. Waston and Crick. Waston and Crick studied proteins, lipids, DNA, RNA only. DNA. Albert Cloud is known as father of cell biology. Father of cell biology, Albert Cloud. Albert Cloud won Nobel Prize in physics, chemistry, literature, physical or medicine, psychology or medicine. Physiology or medicine. Lennon Margulis is associated with Lean Margulis is associated with cell division, DNA structure, endosymbiotic theory, cell theory. Option C. Endosymbiotic theory explain origin of nucleus, origin of mitochondria and chloroplast. C. Cell division. D. DNA replication. P. Origin of mitochondria chloroplast. Lean Margulis published symbiosis theory in 1953, 1974, 1981, 1931. Symbiosis. C. 1981. Protozoa are C. Unicellular. Cork cells observed by Hooke were living dead animal cell, bacterial cells. Dead. Cell biology deals with cells. Simple microscope was used by Hooked, Leeuwenhoek, Russ, Claude. B. Simple microscope was used by Leeuwenhoek. Compound microscope was used by Hook. Nucleus is present in the prokaryotes only, eukaryotes, viruses, all bacteria. Eukaryotes. Protoplasma refers to cell wall, cell membrane, living content of cell, dead part. Living content of cell. Cell theory did not explain origin of cell. 142. Cell division explanation was added by Virchow. DNA is present in nucleus. Electron microscope has high resolution. ఓకేనండి, ఇంతవరకు మనకి బిట్స్ అయినాయండి. ఆ టాపిక్ నుంచి తప్పకుండా లైక్ చేయండి, షేర్ చేయండి, సబ్స్క్రైబ్ చేసుకోండి. కంపల్సరీగా మనకి సెంట్రల్ ఎగ్జామ్స్కి వస్తాయండి బిట్స్. అలాగే టెట్ డిఎస్సి వాళ్ళకి టాపిక్ ఉన్న అందరికి వస్తుంది. ఏపీ తెలంగాణ వాళ్ళకి ఏమైనా స్పెల్లింగ్స్ రాంగ్ చదివితే సారీ అండి. ఓకే, తప్పనిసరిగా లైక్ చేసుకోండి, షేర్ చేసుకోండి, కామెంట్ కూడా పెట్టండి. థ్యాంక్ యూ సో మచ్.